Welcome to the visual notes by Rana Infographics. Our aim is to simplify your learning experience. In our previous video, we talked about the flagellar structure, its motility and pili. In this video, we will talk about the classification of bacteria. The classification of bacteria is based on the following features and characteristics. Based on their mode of nutrition, bacteria can be either autotrophic or heterotrophic. Next, we have classification of bacteria based on their mode of respiration. They are of two types, that is, they can be either aerobic or anaerobic. Next is based on the composition of their cell wall. Bacteria can be divided into two types, which are gram-positive and gram-negative bacteria. And lastly, we have the classification of bacteria based on their shape. Bacteria can be spherical, rod-like, spiral, or comma-shaped. The first criteria is the classification of bacteria based on their mode of nutrition. Let us talk about the first type. Autotrophic bacteria synthesize their own food from inorganic substrates such as carbon dioxide and hydrogen donors. Autotrophic bacteria are further divided into two categories. The first one being photosynthetic bacteria. These bacteria use solar energy for the synthesis of their food and are anaerobic. Next, we have the chemosynthetic bacteria. These bacteria do not have photosynthetic pigment and utilize chemical energy to reduce carbon dioxide to organic food. Next, we have the heterotrophic bacteria. These bacteria do not synthesize their food and are dependent on other organisms or dead organic matter for food. They are further divided into three categories. Parasitic bacteria are bacteria that live on other organisms or hosts and obtain their food from them. Next is the saprophytic bacteria. These bacteria depend upon the dead or organic matters and are major decomposers in nature. And lastly, we have the symbionts. These bacteria live in symbiotic association with other plants for mutual benefits. The next criteria of bacterial classification is based on their mode of respiration. There are two types, the first one being aerobic bacteria. These can survive and grow in an oxygenated environment. Aerobic bacteria are of two types. First one being obligate aerobic bacteria. These are completely aerobic and die in the absence of oxygen. For example, Azotobacter. Second, we have the facultative anaerobic bacteria. These are normally aerobic but can survive in the absence of oxygen. For example, Acetobacter aceti. Next, we have the anaerobic bacteria. These bacteria do not require oxygen for growth and survival. There are two types of anaerobic bacteria. First, we have the obligate anaerobic bacteria. These are completely anaerobic bacteria and they do not have the capacity of aerobic respiration. For example, Clostridium botulinum. Second, we have the facultative aerobic bacteria. These are normally anaerobic but also have the capacity of aerobic respiration. For example, fermentation bacteria except Acetobacter aceti. Next, we have the classification of bacteria based on their cell shape. First, we have the coccus or cocci for plural. These are spherical shaped or elliptical in shape. These are the smallest bacteria. These are maximum resistant bacteria. The cell wall of cocci may be gram-positive with thick peptidoglycan layer or may be gram-negative with thin peptidoglycan layer. For example, Diplococcis pneumoniae. Next we have the Bacillus or Bacilli for plural. These are rod-shaped bacteria. This group includes most of the bacteria. They are the most abundant bacteria. They can form endospores and live for many years in dormancy. They are obligate aerobe or facultative anaerobic bacteria. They are mostly non-parasitic. For example, two parasitic and pathogens are Bacillus anthracis, which causes anthrax, and Bacillus cereus, which causes food poisoning. Next we have the spirillum. These bacteria are spiral or helical in shape. Spirillum bacteria are gram-negative, rigid bacteria having external flagella. Spirochete bacteria have internal periplasmic flagella. These are pathogenic species that cause various serious diseases. For example, Leptospira. Lastly, we have the Vibrium. These are comma-shaped. These are mostly gram-negative bacteria. These cause foodborne diseases. For example, Vibrio cholera. Please note that both cocci and bacilli may occur as single or remain attached in groups after cell division. Those that remain attached can be classified based on their cellular arrangement. Now let us study the classification of bacteria based on cellular arrangement. But first, let us understand what is cellular division of bacteria. When cocci and bacilli divide or reproduce, they create different patterns and shapes. Depending on the plane of cell division, they form varied arrangements. They may occur as single or in groups of 2, 4, 8, etc. Those that remain attached can be classified based on their cellular arrangement. Let us look at the different types of bacteria under cocci. When bacteria exist as a single cell, they are called Monococcus. For example, Micrococcus roseus. Diplococci bacteria occur when cocci are arranged in pairs. Tetrads are arranged in a group of 4 cells. The cell division occurs in 2 different planes. Sarcina are anaerobic gram-positive bacteria. They occur as a group of 8 cells. It is found in the family Clostridiaceae. It is found in the large intestine and skin. Streptococci, here, the cocci are joined in a plane and arranged in a chain pattern. These are non-motile, aerobic and gram-positive bacteria that cause many diseases. Staphylococci. In these bacteria, the cells are arranged in an irregular cluster which looks like grapes. This is due to the division in 3 planes. Now let us talk about the different cellular arrangements in bacilli. Coccobacillus. These are short and oval in shape. They appear like a coccus. Bacillus are single unattached cells that look like a rod. Next we have Diplobacilli. These have 2 rods attached to each other and are found in pairs after cell division. Streptobacilli. Due to cell division in one plane, bacilli are arranged in a chain. Genus Streptobacillus contains gram-negative aerobic or facultative anaerobic bacteria. And lastly, we have the Palisades. After cell division, the bacilli bend and therefore are arranged in a palisade or fence-like structure. Now let us talk about the classification of bacteria based on the composition of their cell wall. A technique called the gram staining technique is used to classify bacteria into 2 groups. Now what is the gram staining technique? In 1882, H.C. Graham developed a staining technique to differentiate between 2 large groups of bacteria based on their different cell wall constituents. The first group is called the gram-positive bacteria. These bacteria have a thick layer of peptidoglycan in their cell walls. When examined under a microscope, they appear purple in color as they retain the crystal violet iodine dye coloring. The second group is called the gram-negative bacteria. These bacteria have a thinner layer of peptidoglycan in their cell walls. When observed under a microscope, they appear pink as they do not retain the crystal iodine dye coloring othering the decoloring process. Now let us understand the gram staining process. The first step is to prepare the slide by drying and fixing the slide. The second step is to stain the bacteria with the crystal violet dye. Next, iodine is added as a mordant, and so a crystal violet iodine complex is formed, so that the dye cannot be removed easily. Next, the bacteria samples are decolorized with alcohol as a result. It dehydrates the peptidoglycan layer, shrinking and tightening it. In this step, we notice that the gram-positive bacteria remain purple since the outer cell membrane remains intact and the crystal violet iodine complex does not get washed off, while the gram-negative bacteria turns white since the outer membrane is degraded and the thinner peptidoglycan layer is unable to retain the crystal iodine complex. In the last step, a counter staining is done with sapphirine, and since it is lighter than the crystal violet, it does not disrupt the purple coloration in gram-positive bacteria, while it turns the gram-negative bacteria red. Now let us see the key differences between the gram-positive and gram-negative bacteria. When washed with alcohol, the gram-positive bacteria remain purple, while the gram-negative bacteria do not retain the color. Gram-positive bacteria are single-layered, while gram-negative bacteria are bilayered. In gram-positive bacteria, the peptidoglycan layer is multilayered and its thickness is about 20 to 30 nanometer. While in gram-negative bacteria, the peptidoglycan layer is single-layered and its thickness is about 8 to 12 nanometer. Gram-positive bacteria have a smooth cell wall, while in gram-negative bacteria, the cell wall is wavy and comes in contact with the cell membrane only at a few loci. Gram-positive bacteria contain 70 to 80% of murine, while gram-negative bacteria contains 10 to 20%. Gram-positive bacteria contains only two flagellar rings, or the S&M rings, which are the inner pair, while in gram-negative all four flagellar rings are found, which are L, P, S and M rings. In gram-positive bacteria, the mesosomes are quite prominent, while in gram-negative bacteria, the mesosomes are less prominent. Only a few pathogenic bacteria are gram-positive, while most pathogenic bacteria belong to the gram-negative group. In gram-positive bacteria, the outer membrane is absent, while in gram-negative bacteria it is present. In gram-positive bacteria, the presence of lipopolysaccharides is virtually none, while in gram-negative bacteria it is high. Gram-positive bacteria primarily produce exotoxins, while gram-negative bacteria primarily produce endotoxins. Some examples of gram-positive bacteria are Staphylococcus, Streptococcus, Pneumococcus, Bacillus, Clostridium, Mycobacterium, Streptomyces. Now let us look at some of the examples of gram-negative bacteria. Salmonella, Pseudomonas, Vibrio, Helicobacter, Haemophilus, Escherichia coli. With that, we complete the section on classification of bacteria. Thank you for watching. If you liked our infographic notes and would like to watch more of such videos, then don't forget to comment, like, share and subscribe to our YouTube channel. If you want to download these notes, then read the description box below and follow us on Instagram.